So we're going to move to the second group of amino acids, which is amino acids that are polar but uncharged. Okay? And there's 6 of them. And I'm just going to explain the term polar. Polar simply means a side chain that can interact with an aqueous environment. That's the simplest way, really, to explain polar. And the most common interaction with an aqueous environment involves forming hydrogen bonds with water. So that's one important feature of polar. Second important feature, which is a good rule of thumb for you, is that a side chain, a polar side chain, will usually have either an oxygen or a nitrogen in the side chain. Oxygen, 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 nitrogen, oxygen. And there's a unique one here that has a sulfur. But as a general rule, the presence of oxygen or nitrogen will tell you that side chain is polar. Okay, so that's an important thing to appreciate. And before I move further, just to kind of tell you what these capital letters are, this is what your classmate asked me about. You don't need to, these are called the amino acid symbols, not abbreviation, symbols, single letter symbols. And as you can see, they're a single letter describing the amino acid. And as you can also see, they don't always match the first letter of the amino acid, because there are many amino acids that start with the same first letter. So anyway, you do not need to know those. Okay? Although they are very important, they are used a lot in depicting protein sequence. That's what's used, is the single letter symbol. But you don't need to memorize these. Just know what they are. Okay, so I'm going to start with the first amino acid here. You still need to know the 3-letter abbreviation. So the first polar side chain is in serine. And actually, if you look at serine, I'm just going to want you to use your imagination. This is actually, if you took this hydroxyl group and put a hydrogen there, that would be alanine. You replace that hydrogen with a hydroxyl, that makes it serine. Okay? So the presence of a hydroxyl group makes serine polar and able to form hydrogen bonds with the aqueous environment. Importantly, these non-polar side chains don't carry a charge. Very important. No negative, no positive charge, just polarity. Second one is threonine. Similar to serine, you just added another methyl, but the hydroxyl is still there. Third one, if you think of this molecule up to here without the hydroxyl, that's phenylalanine. You add the hydroxyl to it, it becomes tyrosine. So tyrosine has a phenol versus phenylalanine as a non-polar amino acid has a phenyl group. So this hydroxyl group makes tyrosine a polar amino acid. So you have 3 hydroxyl in the top 3 amino acids that makes them polar. Then in the bottom, I'll look at asparagine has a carboxyamine, and glutamine has a similar group. They're really very similar. This is slightly longer. So asparagine, glutamine, both have polar side chains because of the carboxyamine, and asparagine the same way. The last one is a very important amino acid called cysteine. And cysteine has a sulfhydryl group, SH, as part of its R group. So CH2SH. This SH is called sulfhydryl. That imparts polarity to cysteine. So this is a unique feature of cysteine. The hydroxyl groups are unique features of serine, threonine, and tyrosine. Asparagine and glutamine have the carboxyamine and group in asparagine and glutamine. Importantly, hydroxy groups, the carboxyamine groups, are polar and are able to form hydrogen bonds with surrounding aqueous environments. I mentioned that already, so that's a very important thing. So tyrosine behaves differently in an aqueous environment than phenylalanine because it doesn't have the hydroxyl group. Phenylalanine is insoluble in an aqueous environment, whereas tyrosine would be soluble. So here's a hydrogen bond sort of representation. I just want you to appreciate one important thing about hydrogen bonds. Hydrogen bonds exist in nature, are abundantly really in nature, and they play very key role in solubility of small and large molecules. In the cell, outside the cell, within the cytoplasm, and so on. Very important. First important thing about hydrogen bond is that you need an oxygen and maybe another oxygen or a nitrogen to share this hydrogen. So you see this is covalently attached in tyrosine. It can form a weak hydrogen bond with another carbonyl group. Or it could be another hydroxyl group. Or it could be another amine group. This bond here is very weak. That's why it's indicated by dashed. It's very weak, nevertheless important. This bond between the oxygen and hydrogen of tyrosine is covalent, very strong. This is non-covalent, very weak. However, hydrogen bonds can be additive. If you have a molecule and it can make 10 hydrogen bonds with its environment, those weak hydrogen bonds have an additive effect on perhaps solubilizing that molecule. Okay? So I want you to appreciate that Hydrogen bonds are very important in solubilizing polar molecules. They usually involve an interaction of hydrogen between that is bonded to an oxygen or a nitrogen and will be interacting with an adjacent oxygen or nitrogen non-covalently, which is this interaction there. And then I'll end with this disulfide bond. Disulfide bond, we said cysteine contains a sulfhydryl group, this SH group. It's a very important amino acid, by the way, in terms of catalysis, enzyme, active sites, and so on. But this sulfhydryl group has the capacity to form what is called disulfide bridges, SS bridges. So here's a cysteine, carboxylic group, amino group, site R group. If you have two cysteines in the vicinity of one another, they can create a covalent bond between them. That's called a disulfide bridge. To form actually a dipeptide. Now, two amino acids called cysteine. This is a pretty strong bond. And this can occur within the same molecule, and it would be called intramolecular disulfide bond, or it could occur between distinct peptides, and it would be called intermolecular disulfide bonds. And you'll appreciate when we get into protein structure that these disulfide bonds play a key role in the tertiary structure of proteins. Okay, so one last thing before, and I'll stop after this. One last thing to remind you about the hydroxyl groups of these three amino acids we talked, the polar amino acids, serine, threonine, and tyrosine. These hydroxyl groups can be covalently modified. What does that mean? It means that you can attach a new group to that hydroxyl group. The most common group that is attached is a phosphate. So you can convert a serine to a phosphoserine, or a threonine to a phosphothreonine, or a tyrosine to a phosphotyrosine. If these residues are part of an enzyme, let's say an enzyme has many serine residues, and you convert them to phosphoserine, you are likely to change the activity of that enzyme because of that conversion. So I want you to appreciate that. We'll talk about it quite a bit later in this block. But these three amino acids can be modified covalently by the addition of a phosphate group. Okay, I think I'll stop here and we'll continue on Friday.